Imagine that we are stepping quietly into a vast and peaceful library, not one filled with paper books or dusty shelves, but a library written in the language of life itself. In this library, every volume is alive. Every page is made of the smallest building blocks of living things. Here, the air is still, the light is soft. There is no rush. Every story waits patiently to be told, even if it has been waiting for millions of years. This is where our journey into genetics begins. Genetics is the gentle science of life's instructions, a study of how traits, characteristics, and patterns are passed from one generation to the next. It is not hurried. It does not shout. It moves at the steady rhythm of growth and renewal, a quiet current flowing through all living beings. To explore genetics is to explore a story that began long before we were born and will continue long after we are gone. If you look at a field of flowers, no two blooms are exactly alike. Some petals are slightly wider, some colors a little deeper, some scents more fragrant. Each variation is a quiet signature, a reflection of the unique genetic instructions each flower carries. These differences are not random accidents. They are part of life's way of ensuring that every living thing is distinct, yet connected to its family. Genetics is found everywhere. It shapes the curve of a bird's wing, the pattern of a snow leopard's spots, the branching of a tree's limbs. It is there in the sound of a voice, the curl of a shell, the precise arrangement of veins in a leaf. Every detail of life, from the simplest bacteria to the tallest redwood, has its roots in the information stored deep within cells. At the heart of genetics is something remarkable: the molecule known as DNA, or deoxyribonucleic acid. DNA is not a lifeless chemical chain. It is the gentle script of existence. You can picture it as a long, spiraling ribbon, twisted into a form so elegant that scientists have compared it to a staircase winding into infinity. It is a structure of symmetry and grace, turning in a calm, steady spiral. If you were to stretch the DNA from just one human cell, it would reach nearly 2 meters in length, yet it is packed so neatly, so delicately, that it fits inside a space invisible to the naked eye. Nature has perfected the art of storage. Like a master librarian, the cell coils, folds, and tucks DNA into organized bundles, protecting it while still keeping it ready to be read when needed. DNA is written in an alphabet of just 4 chemical letters: adenine, thymine, cytosine, and guanine, A, T, C, and G for short. At first glance, it might seem impossible that 4 simple symbols could describe the endless complexity of life. But these 4 letters combine in sequences of incredible variety, forming instructions for building and maintaining every living thing. In their arrangement is the blueprint for feathers and fur, for fins and flowers, for the way your heart beats, and the way your skin repairs itself after a scratch. When we speak of genetics, we are not only speaking about the present moment. We are speaking about deep time, about the passing of life's script from parent to child, generation after generation. Your genetic code is not something you created. It is a gift from your ancestors, carried through countless lives before yours. In every cell of your body is a thread that connects you to a past far beyond memory. Even so, genetics is not only about sameness, it is also about gentle change. Small variations, sometimes tiny changes in the DNA sequence, create differences between individuals. These differences are part of what makes life adaptable, allowing species to adjust to shifting environments over time. In a forest, slight variations in leaf shape may help some plants capture more light. In a pond, tiny differences in a fish's coloring may make it better hidden from predators. In your own family, these changes appear in the smallest of ways, perhaps in the way your smile tilts, or the way your hair curls differently from your parents'. And so genetics is both a preserver and a creator. It keeps the essential blueprint of a species intact, ensuring continuity from one generation to the next. But it also allows for quiet experimentation, testing small variations that may one day become defining traits. It is a balance between stability and flexibility, between tradition and innovation, all playing out in the silent theater of life's code. Every living being carries its genetic story in every one of its cells. And though we often imagine these instructions as purely biological, they are also deeply poetic. They are the poetry of life written in molecules instead of words, a long, unbroken verse stretching from the earliest cells that ever lived to the beings alive today. When you begin to see genetics in this way, you realize it is not merely a field of science. It is a window into connection. The same basic language of DNA runs through all forms of life. It is a reminder that the oak tree outside your window, the songbird in the branches, and you yourself all share a common ancestry, distant yet real. The differences between us are remarkable, but the underlying code is astonishingly similar. In the quiet world of genetics, there is no rush. Change unfolds over years, decades, centuries. A mutation that appears today may not shape a species until hundreds of generations have passed. The transfer of information from parent to offspring is steady and unbroken, like a river flowing through time. And tonight, as we rest here together in this moment of calm, we can begin to see genetics not as a cold set of instructions, but as a gentle, patient storyteller. It tells of beginnings, of continuity, of diversity, and of the endless journey of life itself. In the coming parts of this series, we will move slowly through its chapters. We will learn how this story is written, how it is read, and how it is passed on. We will explore the alphabet of life, the packaging of chromosomes, the way traits move quietly from one generation to the next. We will visit the peaceful gardens of discovery, like Mendel's pea plants, and see how simple observations revealed the patterns still at work in our own time. But for now, it is enough to rest in this opening thought. Within you, and within all living things, there is a quiet, ancient code. It is not rushed, it is not loud, and it is not easily disturbed. It has been whispered through the ages, carried in the cells of every ancestor you have ever had. Tonight, we will listen for that whisper and let it guide us into the soft, steady rhythm of life's most enduring story, the story of genetics. 2. What genetics truly is. If we think of genetics as a story, then part 1 was about opening the first page and finding ourselves in a quiet library of life. Now, as we move forward, we begin to ask the soft question, what is this story really about? What does it mean when we say genetics? The word itself comes from the Greek genesis, meaning origin or beginning, and that is fitting, because genetics is the science of origins, the origins of traits, of patterns, of the small yet meaningful details that make each living being unique. Genetics is not about creating something new from nothing. It is about continuing a thread that has been woven for countless generations, a thread that connects us to all who came before. At its heart, genetics is the study of inheritance, how the instructions for life are passed from parents to their offspring. This passing of instructions happens with quiet precision, as if nature herself were an expert at careful record keeping. In the great flow of life, parents contribute their part of the story, combining it with that of the other parent, and together, these merged instructions form a new individual. This individual will carry not only the marks of its parents, but also the echoes of ancestors stretching far back in time. When you look at a newborn child, a seedling just sprouting from the soil, or a chick emerging from its shell, you are seeing more than a moment of new life. You are seeing a continuation of something ancient, a set of instructions that has traveled quietly, cell to cell, generation to generation. This is the essence of genetics, a living history written in molecules instead of ink. These instructions, which we often call the genetic code, are not visible to the naked eye. They are housed deep within the cells, written in the spiraling structure of DNA. Yet their influence is everywhere. They guide the growth of a tree's leaves each spring. They decide the pattern of veins in a butterfly's wing. They influence the way your hair falls across your forehead, the tone of your voice, and even how your body responds to certain foods. It can be tempting to think of genetics as a fixed and rigid system, a set of unchanging rules. But in truth, genetics is both steady and flexible. The instructions it carries are reliable enough to pass on essential traits, the number of petals on a daisy, the number of chambers in a human heart, yet they are also open to small variations. These variations are what make each individual unique. They allow for the subtle differences between siblings, the slight changes in the leaves of the same tree, the way no two snowflakes are ever exactly the same. One of the gentle truths about genetics is that it is shared. No living thing exists in complete genetic isolation. You share parts of your genetic story with your parents, your grandparents, and all your relatives, but you also share parts of it with other species. The same DNA, letters A, T, C, and G, are used by every living thing on Earth. The way they are arranged is different, but the language is universal. This shared language means that there is a distant but real connection between you and the birds in the trees, the grass beneath your feet, and the fish that swim in faraway oceans. In the peaceful study of genetics, scientists seek to understand not just the structure of DNA, but how it works as a living record. They look at how genes' specific sections of DNA act as instructions for making proteins, the tiny workers that keep our bodies functioning. They explore how genes interact with each other, how they respond to the environment, and how they can change over time. Each discovery adds another thread to the tapestry of understanding, revealing the beauty and complexity of life's design. And yet, genetics is not only about the biological machinery of life. It is also about meaning, the meaning we find in knowing where we come from. Learning about genetics can bring a sense of belonging, a reminder that we are part of a larger story. It shows us that we carry within us the marks of those who lived before, and that we too will pass something on to those who come after us. Think of a meadow in spring. Flowers bloom in colors shaped by genetic instructions, some in pale yellows, others in deep purples. Bees move from blossom to blossom, carrying pollen. The next generation of plants will carry tiny differences, shaped both by their inherited code and by the world in which they grow. Genetics is this same process, happening in every corner of life, from the smallest single-celled organism to the most complex animal. Sometimes, in the quiet moments of study, we realize that genetics is less about control and more about connection. It is a way of seeing the links between past and present, between self and environment, between one form of life and another. It teaches us that we are not isolated individuals, but part of an unbroken flow of life. And just as important, genetics is about potential. The instructions in our DNA are not a strict script dictating every detail of our future. They are more like a set of possibilities, gently shaped by experience, environment, and chance. This is why identical twins who share the same genetic code can still grow into individuals with their own personalities, preferences, and small differences. As we let these thoughts settle in our minds, we can see that genetics is not a cold or mechanical science. It is a quiet, poetic way of understanding life. It tells us that in every living thing, there is both a memory and a promise, a memory of what has been, and a promise of what may yet come. Tonight, as we drift deeper into this story, it is enough to know that genetics is the patient keeper, of life's instructions. It preserves the essential patterns of each species, while allowing for the gentle variations that make life so endlessly diverse. It is the soft voice whispering across time, telling us where we came from, and hinting at where we might go. In the next part of our journey, we will begin to explore the very substance that carries these instructions, the molecule of DNA. We will look at its delicate architecture, its graceful spiral, and the way its simple alphabet gives rise to all the complexity of the living world. But for now, we can rest in this understanding. Genetics is the study of life's quiet script, shared by all living things, patiently passed from one generation to the next, carrying the story of life itself. 3. The story written in every cell. If we were to look closely at ourselves, closer than the eye can see, closer even than a magnifying glass can reveal, we would find a hidden world, quiet and intricate. This world is made up of countless tiny units called cells. They are so small that millions of them could fit on the tip of your finger, and yet each one is a living compartment, carrying with it the full set of instructions for building and maintaining an entire organism. In the gentle study of genetics, it is often said that every cell carries a story. This story is not written in words, but in the arrangement of molecules, the DNA that sits deep within the cell. Whether the cell belongs to a leaf drifting in the breeze, a bird in mid-low flight, or the human body, it carries a complete set of the genetic instructions needed to make that organism. This means that the same story is repeated over and over again, in every cell, like a book, copied perfectly for each reader. Inside your own body are trillions of cells. Most of them are working quietly at this very moment, keeping you alive without any conscious thought from you. A cell in your heart is helping it beat. A cell in your skin is forming a barrier against the outside world. A cell in your stomach is aiding in the digestion of your last meal. And yet, despite their differences in role and location, each of these cells contains the same full genetic script, the same detailed guide for making and maintaining you. The DNA in each cell is stored in the nucleus, a small, rounded compartment often described as the cell's control center. The nucleus is like a secure archive room in a library, where the most important and irreplaceable documents are kept. Here, the DNA is wound carefully into organized bundles called chromosomes, each one holding a specific portion of the genetic story. Together, the chromosomes form a complete set, containing every instruction needed for life. It is a remarkable thing to consider. A cell from your fingertip, a cell from your liver, a cell from your eyelid, all of them carry the same, complete genetic code. What makes them different is not the script they hold, but which parts of that script they are reading and following at a given time. In a way, each cell is like a musician with the same sheet music in front of them, choosing which notes to play depending on the role they have in the symphony of the body. In plants, the same truth holds. A leaf cell and a root cell in a single tree both contain the full genetic story of that tree. Yet the leaf cell focuses on reading the parts of the script that help it perform photosynthesis, the process of turning sunlight into food. The root cell reads a different set of instructions, ones that guide it to take in water and minerals from the soil. It is this selective reading of the genetic script that allows a single organism to have so many different kinds of cells, each perfectly adapted to its job. The repetition of this story in every cell is one of the quiet marvels of life. It ensures that no matter where a new cell forms in the growing tip of a branch, in a healing wound, or in the developing embryo, it has the complete set of instructions to know what it is, where it belongs, and how to function. Even in the smallest of life forms, like bacteria, the principle remains. A single bacterial cell contains all the genetic information it needs to grow, reproduce, and respond to its environment. While it may be only a fraction of the size of a human cell, Its DNA is still the keeper of a living history, connecting it to generations of bacteria before it. When you realize that every cell in your body carries your full genetic story, you begin to see yourself in a new way. You are not simply one being, but a vast community of cells, each one carrying your history. And within each of those cells lies a record, a book of instructions that began writing itself long before you were born. This book was written, in part, by your parents, and before them, by their parents. It is a story that extends backward, unbroken, for countless generations. The idea that your genetic story is written in every cell also brings a sense of resilience. A single cell, if cared for in the right way, holds enough information to rebuild the blueprint of the whole organism. This is why certain animals, plants, and even scientific experiments can grow a new individual from just a single cell. The story is all there, complete, and waiting in the tiniest living space. And yet, for all its importance, this story is fragile. DNA can be damaged by harsh chemicals, radiation, or errors during the process of cell division. Cells work constantly to repair and protect their DNA, much like a careful librarian who mends the pages of a treasured manuscript. Without this protection, the story could become unreadable. Fortunately, life has spent billions of years perfecting ways to keep the code intact. There is something comforting in knowing that every cell, no matter how small or hidden, carries the same complete identity of the organism it belongs to. Whether it is a cell deep in the root of a tree, or one at the tip of a leaf, a cell in your heel, or one in your lips, the entire genetic story is present and whole. It is a quiet reassurance, a reminder that your identity is written not just once, but trillions of times throughout your body. In this way, the body is not a single copy of a story, but a vast library filled with the same treasured book over and over again. If one page becomes worn, another copy exists. If one copy is lost, the story is still safe in countless others. This is one of the deep, patient safeguards of life. As we think about this, we begin to understand why genetics is such a profound science. It is not only about molecules and chemistry. It is about memory, continuity, and identity. It is about the way life holds onto its own instructions and repeats them faithfully, generation after generation, cell after cell. So as we rest in this thought, we can picture ourselves as a vast collection of quiet storytellers, each one holding a complete copy of the tale of our being. They carry our history, our potential, and our connection to all that has lived before. And in the next part of our journey, we will look more closely at the substance of this story, the nature of DNA itself, the delicate yet enduring thread that carries the code of life. 4. The nature of DNA. Imagine for a moment a thread so fine that it cannot be seen with the naked eye. This thread is wound and coiled in the tiniest of spaces, tucked away in the quiet heart of every cell. Yet within its delicate structure lies the complete blueprint for building and maintaining life. This thread is DNA, deoxyribonucleic acid, the gentle molecule that carries the instructions for all living things. When scientists first discovered DNA, they could not have imagined the beauty of its form. It was only in the middle of the 20th century that its shape was revealed, a double helix, spiraling upward like a staircase made for something smaller than the smallest grain of sand. Each side of this staircase is made from long strands of sugar and phosphate molecules, strong enough to protect the precious code within. Connecting the two sides are pairs of chemical letters called bases: adenine, thymine, cytosine, and guanine. These bases always pair in the same way: adenine with thymine, cytosine with guanine. It is a pairing so consistent, so dependable, that it has been called the base pairing rule. These pairs are like rungs on the staircase, each rung holding the sides of the helix together, while also forming the steps by which the code of life can be read. The sequence of these bases is what forms the genetic instructions. In the same way that letters combine to make words and words combine to make stories. These chemical letters combine to form the language of life. And though the alphabet has only 4 letters, the variety of possible arrangements is beyond counting. This is how the DNA in a tree can give it leaves shaped for catching sunlight, and the DNA in a bird can give it wings made for flying. The length of DNA is astonishing. If you could stretch the DNA from just one human cell into a straight line, it would measure nearly 2 meters long. And yet, inside the cell's nucleus, it is coiled so tightly and precisely that it fits into a space smaller than a speck of dust. This careful folding is an art perfected by life itself. The DNA wraps around tiny proteins called histones, which act like spools, keeping the long threads neat and organized. Without this coiling, the delicate strands could become tangled or damaged. DNA is also remarkably stable. It can survive long after a cell has died, preserved in ancient bones or frozen remains. From such DNA, scientists have learned about creatures that lived tens of thousands of years ago. Yet, despite its stability, DNA is also flexible enough to be copied and read again and again. Every time a cell divides, its DNA must be copied so that each new cell has the same complete set of instructions. This copying process is called replication, and it is carried out with extraordinary accuracy, guided by enzymes that move along the strands, reading and matching each base with its partner. But even with such careful work, changes can occur. Sometimes, a base is paired incorrectly, or a small section of DNA is copied twice, or a piece is skipped altogether. These changes are called mutations. Many mutations have little to no effect. Some can be harmful, while others can be beneficial, offering the organism a small advantage in its environment. Over long spans of time, these changes can shape the direction of a species, like gentle currents guiding the course of a river. One of the most remarkable things about DNA is that it is universal. Every living thing on Earth, from the smallest bacterium to the tallest tree, uses DNA to store its instructions. The code itself is written in the same 4 letters, A, T, C, and G. What differs is the sequence, the order in which those letters appear. In this way, all life is connected, branches of the same great tree, growing from the same root. The universality of DNA means that there is a deep and ancient kinship between all living beings. The DNA in your cells shares similarities with the DNA in a whale, a hummingbird, a sunflower, even the tiniest of fungi. This shared code tells us that life, in all its diversity, has a common origin. The differences we see today are the result of millions upon millions of years of changes in the sequence of those 4 letters, slowly shaping life into the countless forms we know. Inside the cell, DNA does not simply sit idle. It is constantly being read and used. Sections of DNA, called genes, contain the specific instructions for making proteins, the molecules that carry out nearly every function in the body. Some proteins build structures, like the keratin in your hair and nails. Others act as enzymes, speeding up chemical reactions that would otherwise be too slow to sustain life. Still others serve as messengers, carrying signals between cells and organs. The flow of information from DNA to proteins is sometimes called the central dogma of molecular biology. It begins when the DNA code is copied into a similar molecule called RNA, in a process known as transcription. The RNA then carries the code to the ribosomes, tiny factories inside the cell, where proteins are assembled according to the instructions. In this way, the quiet script of DNA is brought to life in the form of active molecules that keep the organism alive and functioning. What makes DNA so extraordinary is not just the precision of its design, but the elegance with which it combines simplicity and complexity. 4 chemical letters, arranged in countless ways, are enough to describe every form of life on this planet. And these instructions are stored not in a library that must be visited, but within the very fabric of life itself, carried in every cell, read and reread without end. As we think about the nature of DNA, it is hard not to feel a sense of quiet awe. This molecule, so small and delicate, holds the story of life. It is both ancient and ever new, shaped by the deep past, yet actively shaping the present. It is the link between parents and children, between ancestors and descendants, between all living things. In the stillness of this thought, we can imagine each of our cells cradling its DNA like a treasured manuscript, carefully preserving it through every moment of our lives. And when life passes from one generation to the next, it is this manuscript copied, shared, and sometimes gently altered that continues the story. In the next part of our journey, we will look at the 4 chemical letters of DNA more closely, the nucleotides, and see how this simple alphabet forms the endless variety of life's instructions. But for now, we can simply rest in the knowledge that DNA is the quiet architect of life, working patiently in every cell, every moment, without pause. 5. The alphabet of life. Nucleotides. If you have ever held a book in your hands, you know that it is made from words, and words are made from letters. The letters themselves are simple, just shapes on a page, yet when arranged in certain ways, they carry meaning. They tell stories, pass on knowledge, and preserve history. Life, too, has its own alphabet. It is not written with ink on paper, but with chemicals arranged in a precise and beautiful pattern. These are the nucleotides, the 4 letters of the genetic code. A nucleotide is a small, repeating unit that makes up DNA. Each one is like a bead on a string, and together they form the long, winding thread of the DNA molecule. Though each nucleotide shares the same general structure, it has a unique base, a chemical group that gives it its identity. There are 4 types of these bases: adenine, thymine, cytosine, and guanine. In the language of genetics, they are simply called A, T, C, and G. At first, it might seem hard to believe that just 4 different chemical letters could write the instructions for every living thing on Earth. And yet, this is exactly how life works. These 4 nucleotides, arranged in countless different combinations, are enough to describe the color of a flower, the song of a bird, the shape of a mountain lion's paw, and the curl of a human smile. The beauty of this system lies in its simplicity and its consistency. Across all of life, A always pairs with T, and C always pairs with G. This pairing is like the way puzzle pieces fit together. Only the right shapes can match. These base pairs form the rungs of the DNA double helix, holding the 2 strands together. When the DNA is copied or read, the cell uses this pairing rule to ensure accuracy. If there is an A on one strand, the cell knows there must be a T on the other. If there is a C, there must be a G. Each nucleotide is made of 3 parts. First, there is a sugar molecule, shaped like a gentle pentagon, which forms the backbone of the DNA strand. Attached to the sugar is a phosphate group, linking one nucleotide to the next, like links in a chain. And finally, the base A, T, C, or G is attached to the sugar, reaching inward to connect with its complementary partner on the other strand. This elegant structure repeats over and over again, like the rhythm of a soft song, until an entire chromosome is formed. It is easy to imagine DNA as a static thing, a fixed chain of nucleotides lying quietly in the nucleus, but in reality, it is dynamic. The sequence of nucleotides is constantly being read and sometimes copied. Enzymes glide along the strands like careful readers, checking each letter and matching it to its partner. The order of these letters, their sequence, is what matters most. Just as rearranging the letters in a word changes its meaning, changing the sequence of nucleotides can alter the meaning of the genetic instructions. Sometimes these changes are small and go unnoticed. Other times, they can have profound effects, shaping traits in new ways. A single change in a nucleotide sequence can be enough to influence the color of a flower's petals or the way a protein folds inside the body. Over time, changes in nucleotide sequences contribute to the diversity of life, creating variations that may help a species adapt to its surroundings. What is perhaps most astonishing about nucleotides is their universality. No matter where you look in the cells of a towering oak tree, in the scales of a tiny lizard, in the wings of a butterfly, you will find the same 4 nucleotides. The code is shared by all life. The differences between species come not from the letters themselves, but from the way they are arranged and the length of the sequences they form. The sequence of nucleotides in a gene is like a sentence. The gene for eye color, for example, may contain thousands of nucleotides in a precise order. The cell reads this sequence and translates it into a protein that influences pigment production. If even one letter in that sequence changes, it can alter the protein and change the shade of the eye's color. In this way, the smallest changes in the genetic alphabet can have visible effects in the real world. And yet, for all their importance, nucleotides are incredibly small. Billions of them fit into the DNA of a single cell, arranged in a perfect, orderly fashion. They are invisible to the naked eye, but their effects are everywhere. Every feature of every organism, from the graceful stretch of a giraffe's neck to the spiral shell of a nautilus, is built from instructions written in this simple 4-letter code. It is worth pausing to think about the elegance of this system. Imagine if every story in the world, every poem, every piece of music, could be written with just 4 letters. That is what life does. With A, T, C, and G, it writes the entire book of nature. These letters have been in use for billions of years, passed down from the earliest forms of life to everything that lives today. They are the most enduring alphabet in existence. Even now, in this very moment, the nucleotides in your cells are doing their work. They are quietly holding your genetic information, ready to be read when needed. They are copied with extraordinary care each time a cell divides, ensuring that the instructions are preserved. And they connect you, in a deep and fundamental way, to every other living being that has ever existed. In the stillness of this thought, we can see nucleotides for what they truly are: the simplest and yet most powerful letters ever written. They form the bridge between chemistry and life, between the physical structure of a molecule and the unfolding beauty of a living organism. They are constant, dependable, and universal, the quiet keepers of life's language. In the next part of our journey, we will turn from the letters themselves to the shape of the book they form. We will explore the double helix, the graceful spiral staircase that holds the nucleotides in place and protects the instructions they carry. But for now, we can rest in the gentle understanding that life's entire story begins with just 4 letters, repeating endlessly, writing the world around us. 6. The spiral of the double helix. If you could shrink yourself down, far smaller than the smallest grain of sand, and drift inside a cell, you would eventually find yourself in a quiet, sheltered space, the nucleus. And there, resting like a coiled ribbon in a treasure box, you would see something extraordinary: the spiraling form of DNA. This shape is called the double helix. It is both simple and intricate, ancient yet perfectly suited to its purpose. 2 long strands of nucleotides wind around each other in a gentle, regular twist. Together, they look like a staircase that rises into the distance, except instead of solid steps, each rung is made from a pair of chemical bases: A paired with T, C paired with G. The symmetry is elegant, almost musical, each pairing repeating without flaw. The 2 strands of DNA are not identical. They are complementary. Where one has an A, the other has a T. Where one has a C, the other has a G. This complementarity is what allows DNA to be copied with such accuracy. When the molecule needs to be duplicated, the 2 strands separate, and each serves as a template for making a new partner strand. It is as if nature designed a book that could always make a perfect copy of itself, simply by opening its pages. The twist of the helix is gentle and even, about 10 base pairs per full turn. This winding shape is not just beautiful, it is efficient. The spiral makes the molecule compact, allowing vast lengths of DNA to fit into the tiny space of a cell's nucleus. Without this twist, the DNA would be far too long and fragile to be stored and used effectively. Each strand of the double helix is made from a strong backbone of sugar and phosphate molecules. This backbone forms the outer edges of the spiral, providing stability and protecting the delicate base pairs inside. The bases themselves are stacked in the center, like the steps of that tiny spiral staircase, held together by gentle hydrogen bonds. These bonds are strong enough to keep the structure stable, yet weak enough to be broken when the DNA needs to be read or copied. The double helix is not rigid. It can bend, coil, and even loop back on itself. This flexibility allows the DNA to be folded into complex shapes so that it can be stored neatly. In human cells, the DNA is wrapped around proteins called histones, forming compact bundles called nucleosomes. These bundles coil further, winding into even tighter structures until the DNA is condensed into chromosomes. The discovery of the double helix in 1953 was a turning point in science. It was the moment when the abstract idea of a genetic code became a tangible structure, something that could be seen, measured, and understood. Scientists James Watson and Francis Crick, building on the work of Rosalind Franklin and Maurice Wilkins, revealed this spiral form to the world. In doing so, they gave us a clear vision of the physical structure that carries the blueprint of life. But the double helix is more than just a scientific model. It is a symbol of connection and continuity. Its two strands are bound together, supporting and completing each other. This relationship mirrors the way genetic information is passed down not as a single strand, but as a union of complementary parts, each dependent on the other. Within the spiral of the double helix lies the code that shapes everything a living organism becomes. The sequence of bases along its length determines which proteins will be made and in what amounts. These proteins then build and maintain the organism, influencing traits as visible as eye color and as hidden as the enzymes that digest food. The twist of the helix also plays a role in how the DNA is read. Certain sequences of bases form recognition sites for proteins that bind to DNA, marking where genes begin or end. The spiral shape allows these recognition sites to be positioned in a way that makes them accessible to the cell's molecular machinery. In this way, the architecture of the double helix is inseparable from the way the genetic code is used. If you could follow a single DNA double helix from end to end, you would trace an unbroken path through the entire history of an organism's genetic information. Along the way, you would pass through countless genes, regulatory regions, and stretches of non-encoding DNA that may serve as quiet archives of evolutionary history. Every twist, every rung, is part of the story. And yet, for all its complexity, the double helix is a humble structure. It works silently, without drawing attention to itself. It does not announce its importance, but without it, life as we know it could not exist. The spiral staircase of DNA is the foundation on which all living things are built. There is also a quiet poetry in the fact that the double helix is not unique to one form of life. The same spiraling shape is found in bacteria, plants, animals, and humans alike. It is the shared architecture of life, a design that has endured for billions of years because it works so perfectly. When you think of the double helix, you might imagine it as a single molecule, but in truth, each of your trillions of cells contains many copies of this spiral, each one holding the entire genetic code for your body. Every copy is identical, a perfect reproduction of the original, faithfully passed along as cells divide. In the stillness of this thought, the double helix becomes more than just a structure. It is a quiet, continuous thread winding through time and space, linking you to your ancestors and to every other living thing. It is the elegant curve of life's handwriting, looping endlessly in the script of nature. In the next part of our journey, we will turn our attention to the genes themselves, the sections of DNA that carry the specific instructions for building the many parts of a living organism. But for now, we can rest with this image in our minds: a spiral staircase, rising and turning forever, carrying the code of life in its graceful twist. 7 genes, life's small instructions. Inside the long spiraling molecule of DNA, there are regions that hold particular significance. While the entire DNA strand is important, certain stretches carry specific, actionable instructions. These are the genes, the small, carefully arranged sequences that tell the cell how to make the proteins and molecules it needs to function. A gene is not a large part of the DNA. In fact, compared to the full length of the DNA molecule, a gene is like a short paragraph in an enormous book. But even though a gene may seem small in size, it can have a profound influence on how an organism develops and lives. Each one contains the code for a particular protein, which will have its own role to play in the body. Proteins are the builders, messengers, and caretakers of the cell. Some form the scaffolding that holds the cell together. Others carry oxygen through the bloodstream, or detect light in the eye, or break down food into usable energy. Each protein has a shape and function determined by the exact sequence of nucleotides in its corresponding gene. This means that the recipe for a protein is written directly into the DNA, waiting to be read when needed. When a cell needs a particular protein, it opens the section of DNA containing the gene for that protein. The sequence of nucleotides in the gene is copied into a molecule of RNA in a process called transcription. RNA is like a working copy of the instructions. It can travel out of the nucleus to the ribosomes, the cell's protein-building factories. There, the RNA is read, and the protein is assembled one building block at a time. The proteins built from genes are not just static structures. They are active participants in nearly every process that sustains life. Without them, cells could not grow, repair themselves, or communicate. And because proteins carry out such a wide variety of roles, the genes that encode them are vital to every aspect of an organism's existence. In humans, scientists estimate there are about 20, 0 to 25, genes. Each one is a small instruction within the larger manual of the genome. Some genes are active all the time, producing proteins that are constantly needed, like those involved in energy production. Others are turned on only in certain cells, at certain times, or in response to specific signals from the environment. The pattern of which genes are active and when they are used is called gene expression. This pattern is not random. It is carefully controlled, ensuring that each cell in the body produces the right proteins for its particular function. For example, a muscle cell and a nerve cell both have the same DNA, but their gene expression patterns are different. The muscle cell activates genes for making proteins that contract fibers, while the nerve cell activates genes for transmitting electrical signals. Sometimes, the activity of a gene can be influenced by the environment. This means that even though the DNA sequence remains the same, external conditions can change how a gene is expressed. This is part of the delicate balance between genetics and environment, a relationship we will explore more deeply later in our journey. It is important to remember that genes do not work in isolation. They interact with one another, often in complex ways. A single trait, such as height or skin tone, is usually shaped by the combined influence of many genes working together, along with environmental factors. And some genes can affect more than one trait, weaving a web of connections throughout the organism. Not every part of the DNA is a gene. In fact, much of the DNA in a genome does not directly code for proteins. Some of these non-coding regions help regulate the activity of genes, acting as switches that turn them on or off. Others may serve as structural support for the DNA, or as remnants of ancient genetic material from long-past ancestors. Even though they are not genes themselves, these regions still play a role in how the genetic story is told. Genes are also the carriers of inherited traits. The version of a gene you receive from your parents can shape aspects of your appearance, your physiology, and even some of your tendencies. These versions of a gene are called alleles. For example, the gene that influences eye color comes in different alleles, some leading to brown eyes, others to blue, green, or other shades. Which alleles you inherit will influence the color of your eyes, though other genes and environmental factors may also play a part. Because genes are passed down from one generation to the next, they form a link between the past and the present. The genes you carry today are a blend of those carried by your parents, your grandparents, and countless ancestors before them. This genetic inheritance is part of what makes you who you are. It is a quiet, continuous thread connecting you to those who came before, and, one day, to those who will come after. In nature, the passing of genes is not limited to parents and offspring. In some microorganisms, genes can be exchanged directly between individuals, even between different species. This sharing of genetic information allows for rapid adaptation and change, helping life survive in shifting environments. It is one more example of how genes are not just static instructions, but living elements of a constantly evolving story. When we step back and look at the bigger picture, genes are like individual notes in the great symphony of life. Alone, a single note might seem small, even insignificant, but when combined with thousands of others, each precisely timed and tuned, the notes become a melody one that has been playing for billions of years. In the next part of our journey, we will look at how DNA is organized into chromosomes, the careful packages that protect and store our genetic material. But for now, we can rest in the understanding that genes are life's small instructions, compact, precise, and endlessly important. They are the quiet guides that shape the countless forms and functions of living things, from the smallest bacterium to the largest whale, from the first breath of a newborn child to the turning of leaves in autumn. 8. Chromosomes. DNAs. Careful packages. Inside each cell, DNA is both incredibly long and incredibly delicate. If we could take the DNA from a single human cell and stretch it out end to end, it would measure close to 2 meters in length. And yet, it must fit into a space so small it cannot be seen without a microscope, the nucleus, which is only a few millionths of a meter across. To make this possible, DNA is stored in a way that is both compact and protective. This careful packaging is what we call a chromosome. A chromosome is not just a random bundle of DNA. It is a precisely organized structure designed to keep the genetic material safe and in order. The DNA wraps around small spool-like proteins called histones. These histones help coil the DNA tightly without tangling it. Imagine winding a long, delicate thread around a series of tiny spools, then stacking those spools neatly so they take up as little space as possible. This is what happens inside your cells, every moment of your life. Once wrapped around histones, the DNA and protein together form a structure called chromatin. Chromatin can be loosely packed when the cell needs to read the DNA, or tightly packed when the DNA needs to be stored safely. This flexibility allows the cell to protect its genetic material while still keeping it accessible for important processes like making proteins or copying the DNA before cell division. When a cell is preparing to divide, the chromatin coils even more tightly, forming the familiar X-shaped structures that we often see in textbook illustrations of chromosomes. Each X shape is made of two identical halves called sister chromatids. connected in the center at a point called the centromere. These identical halves are the result of the DNA being copied during the cell cycle, ensuring that each new cell will receive a complete set of genetic instructions. Humans have 46 chromosomes in most of their cells, arranged in 23 pairs. One chromosome in each pair comes from the mother, and the other comes from the father. These pairs carry matching sets of genes, though the versions of the genes, the alleles, may differ. This pairing system helps preserve the stability of our genetic information across generations while also allowing for variation. The number of chromosomes is not the same for all species. A fruit fly has just 8 chromosomes, while some species of ferns can have more than 1,000. The number alone does not determine the complexity of the organism. Rather, it is the sequence of the DNA, the order of the nucleotides, and the way the genes are arranged and regulated that shapes each species. Chromosomes serve another vital role. They protect DNA from being damaged or lost during cell division. Without chromosomes, the long, fragile strands of DNA could easily break or become tangled, leading to errors in the genetic code. By keeping DNA neatly packaged, chromosomes act as both guardians and organizers of the genetic story. At the ends of each chromosome are special regions called telomeres. Telomeres are often compared to the plastic tips on the ends of shoelaces, which prevent the lace from fraying. In the same way, telomeres protect the ends of DNA from damage. Each time a cell divides, its telomeres become slightly shorter. Over time, this shortening plays a role in the aging process, and when telomeres become too short, the cell can no longer divide effectively. The organization of DNA into chromosomes also makes it possible for cells to pass on genetic information with remarkable accuracy. During reproduction, chromosomes are carefully sorted and divided so that each new cell, whether it's part of a growing embryo or simply replacing an old cell in your body, receives a complete set of instructions. In sexual reproduction, the chromosomes from both parents combine, blending genetic traits in unique ways to form the genetic blueprint of a new individual. Sometimes, changes in the structure or number of chromosomes can occur. These changes can happen naturally and may have no noticeable effect, or they can lead to significant differences in how the body develops and functions. In some cases, chromosomal changes contribute to the diversity of life, while in others, they can cause genetic disorders. Nature has many systems in place to maintain the integrity of chromosomes, but like all living processes, it is not entirely free from variation. When we step back and consider the chromosome as a whole, it becomes clear that it is more than just a container for DNA. It is an intricate system, balancing the need for storage, accessibility, and protection. It keeps the vast library of genetic information safe, while allowing the cell to open the books whenever it needs to read or copy them. In every living thing, chromosomes are the quiet keepers of the genetic archive. They do their work silently, without recognition, yet their role is essential. Without them, the genetic instructions would be vulnerable and chaotic. With them, the story of life is preserved in perfect order, ready to be passed along to the next generation. We can imagine them as rows of scrolls in a perfectly organized library, each one containing the full story of the organism. The scrolls are stored carefully, yet they can be unrolled and read when the time is right. This is the harmony of the chromosome, the balance between keeping the code safe and keeping it usable. And so, the chromosome is not just a structure of science. It is a symbol of life's meticulous care for its own continuity. It reminds us that even at the smallest scales, nature works with precision and intention. The packaging of DNA is not random. It is an act of protection, preservation, and respect for the story it carries. In the next part of our journey, we will follow that story as it moves from one generation to the next. We will explore inheritance, the quiet, steady passing of genetic information from parent to offspring. This is how the story written in chromosomes is carried forward, linking the lives of ancestors with the lives of their descendants in an unbroken chain. But for now, we can rest with the image of these careful packages, holding within them the blueprint of life, coiled in perfect order, safe in the quiet center of the cell. 9. The beauty of variation. If you were to wander through a wildflower meadow in late spring, you would see an endless array of colors, shapes, and patterns. Some blossoms might be deep crimson, others soft yellow, and still others a delicate shade of violet. Even flowers of the same species will often differ subtly, perhaps in the way their petals curl or in the tone of their fragrance. This quiet diversity is one of the most beautiful truths of nature. It is the result of variation, the natural differences in traits that occur between individuals of the same species. Variation is not a flaw in nature's design. It is one of its greatest strengths. Without variation, all members of a species would be identical. There would be no differences to allow some individuals to thrive in changing conditions while others might struggle. Variation is what gives life flexibility, adaptability, and resilience. It is the gentle yet powerful way that nature prepares for an uncertain future. At the heart of variation lies genetics. Even though all individuals of a species share the same set of genes, the versions of those genes, the alleles, can differ. These differences may be as small as a single change in the sequence of nucleotides, or they may involve more complex rearrangements of genetic material. Sometimes, these differences are inherited from parents. Other times, they arise as new changes in the DNA, called mutations. Not all variations are visible. Some are obvious, like differences in eye color, height, or leaf shape. Others are hidden within the workings of the body, variations in metabolism, immune system strength, or the way cells respond to certain signals. These hidden differences can be just as important as the visible ones, quietly shaping an individual's chances of survival and reproduction. Mutations, though they might sound alarming, are a natural and ongoing part of life. They occur whenever DNA is copied, whether in the making of new cells or in the passing of genetic information to the next generation. The cell has remarkable systems for checking and correcting mistakes, but small changes still slip through. Some mutations may have no noticeable effect at all. Others can be harmful, but some can be beneficial, offering a trait that helps an organism live more successfully in its environment. Over many generations, these beneficial variations can spread through a population, shaping the direction of evolution. Variation also comes from the mixing of genetic material during sexual reproduction. When sperm and egg cells are formed, they each receive a shuffled combination of the parent's chromosomes. This shuffling, called recombination, ensures that no two offspring are genetically identical, except in the rare case of identical twins. It is this blending of parental traits, along with the small, random changes that occur naturally, that produces the infinite variety we see within each species. We can see variation everywhere. In a litter of kittens, each one has its own unique coat pattern, eye color, and personality. In a forest of maple trees, no two have identical arrangements of branches and leaves. Even among humans, the differences in facial features, voices, and mannerisms are endless. This variety is part of what makes life rich and interesting. Variation also serves as nature's insurance policy. Imagine a sudden change in the environment, a shift in climate, the arrival of a new predator, or the outbreak of a disease. If all individuals in a species were identical, such a change might threaten the entire population. But with variation, some individuals are more likely to have traits that help them survive the challenge. These individuals can pass their traits to the next generation, helping the species adapt and endure. Some variations are the result of long adaptation to specific environments. For example, polar bears have thick white fur, perfect for blending into snowy landscapes, and retaining heat in frigid temperatures. Brown bears, their relatives in warmer regions, have darker fur and different body proportions suited to their environment. Though they share much of the same genetic blueprint, variations in certain genes give them traits that fit their surroundings. Variation is not only functional, it is also a source of beauty. The diversity of patterns in a butterfly's wings, the range of colors in autumn leaves, the subtle differences in birdsong, all of these are products of variation. They enrich our world, reminding us that nature values variety as much as it values order. Humans, too, are part of this story. The differences between people in appearance, in abilities, in perspectives, are rooted partly in variation within our genes. These differences shape cultures, innovations, and ways of life. They allow communities to draw on a wide range of strengths, making societies more adaptable and creative. It is also worth noting that variation works hand in hand with the environment. A trait that is advantageous in one setting may be less helpful in another. Dark fur may help an animal stay hidden in a forest, but it might stand out in a snowy field. Long legs may be perfect for running across open plains, but less useful in dense undergrowth. This interplay between variation and environment is one of the quiet forces that shape the course of life over time. When we see variation in its full context, it becomes more than just a biological concept. It becomes a reminder that differences are not only natural, but essential. Variation ensures that life is never static, that it is always experimenting, always exploring new possibilities, always ready for what might come next. In the next part of our journey, we will meet one of the first scientists to carefully observe these patterns of inheritance and variation, Gregor Mendel. His quiet experiments in a garden of peas revealed the basic rules by which traits are passed down, offering us a first glimpse into the underlying patterns of genetics. But for now, we can simply rest in the awareness that the world's diversity in all its colors, shapes, and forms is the gift of variation. It is nature's way of keeping life strong, adaptable, and endlessly beautiful. 10. Mendel's Garden of Discovery. Long before the structure of DNA was known, before microscopes could reveal the intricate details of chromosomes, there was a man quietly working in a monastery garden. His name was Gregor Mendel, and he had no way of knowing that his patient experiments would one day form the foundation of modern genetics. Mendel's tools were simple: a garden of pea plants, a small notebook, and the careful, steady hands of a man who observed nature with great patience. The monastery in which Mendel lived was surrounded by orderly rows of plants. It was here, in this peaceful setting, that he began to notice patterns in the way traits appeared in successive generations of pea plants. He saw that certain characteristics like flower color, seed shape, and plant height seemed to follow specific rules when passed from parent plants to their offspring. Mendel chose pea plants for his studies not by accident, but because they had traits that were easy to see and easy to control. Their flowers could be carefully opened and pollinated by hand, allowing him to decide exactly which plants would be crossed with one another. By controlling these pairings, Mendel could track how traits appeared in each new generation. He began by selecting plants with opposite traits, for example, one plant with purple flowers and another with white flowers. When he crossed them, the next generation, which he called the first filial generation or phone, all had purple flowers. The white flower trait seemed to have disappeared, but when he allowed these phone plants to self-pollinate, the following generation, the F2, revealed the white flowers again, appearing in a consistent ratio alongside the purple flowers. This was no coincidence. Mendel repeated his experiments many times, not only with flower color, but with other traits such as seed shape round versus wrinkled and plant height tall versus short. Each time, the patterns reappeared, as regular as clockwork. Through his careful record keeping, Mendel realized that traits are determined by discrete units, which he called factors. Today, we know these factors as genes. Mendel observed that each plant carried two copies of each factor, one from each parent. In some cases, one version of the factor could mask the other. This is what happened with the purple and white flowers. The factor for purple flowers was dominant, meaning it would show in the plant even if only one copy was present. The factor for white flowers was recessive, meaning it would only appear if both copies were white. From these observations, Mendel formulated two important principles. The first was the law of segregation, which states that the two factors for a trait separate during the formation of reproductive cells, the pollen and the ovules, so that each parent contributes only one factor to their offspring. The second was the law of independent assortment, which states that factors for different traits are passed on independently of each other, so the inheritance of one trait does not necessarily affect the inheritance of another. In his time, Mendel's work was not widely recognized. The scientific community of the mid-1800s did not yet have the tools or understanding to see how his observations fit into the broader picture of biology. His studies were quietly published and then largely forgotten for decades. It wasn't until the turn of the 20th century, long after Mendel's death, that scientists rediscovered his experiments and realized their significance. Today, Mendel is remembered as the father of genetics. His pea plants, his patient note-taking, and his ability to see patterns in the natural world laid the groundwork for our modern understanding of inheritance. His work taught us that traits are not blended in some vague fashion, but are passed along as distinct units, preserved across generations. What makes Mendel's story so remarkable is its simplicity. He did not need complex equipment to uncover the rules of inheritance. He needed only patience, careful observation, and a mind attuned to the patterns of life. In a quiet garden, with nothing more than rows of pea plants and the changing seasons, he revealed a truth that connects every living thing, the truth that traits follow laws, that inheritance has order, and that variation, though unpredictable in appearance, is shaped by consistent principles. Even today, the lessons from Mendel's garden are taught to students around the world. His experiments remind us that science often begins with simple questions. What happens if I cross these two plants? Why do some traits appear while others seem to vanish? And what might happen in the next generation? As we picture Mendel walking among his plants, gently tending to blossoms, collecting seeds, and noting the results in his ledger, we can feel the unhurried pace of his work. It was not driven by deadlines or pressure. It followed the rhythm of the growing season, the opening of flowers, the ripening of pods. His discoveries came slowly, like the unfolding of the plants themselves. In many ways, Mendel's work is a reminder that the deepest insights often come from quiet observation. It shows us that the laws of genetics, which now seem so precise and formal, were first glimpsed in the gentle pattern of petals and pods. And it reminds us that even the most important scientific truths can begin as seeds, small, unassuming, and planted in the soil of curiosity. In the next part of our journey, we will look more closely at what Mendel discovered about dominance and recessiveness, and how these principles shape the way traits appear in living things. But for now, we can linger in the peaceful image of a monastery garden, where the wind moves softly through rows of pea plants, and where the patient work of a single man quietly revealed the enduring patterns of life. 11 patterns in the garden, dominant and recessive traits. When Gregor Mendel walked among his rows of pea plants, he noticed something curious. Certain traits seemed to appear more often, even when he crossed plants with different characteristics. It was as though some traits were bold, stepping forward to be seen, while others waited quietly in the background. Over time, Mendel realized that this was not random. These patterns followed consistent rules, the principles of dominance and recessiveness. Dominant traits are those that appear whenever at least one copy of the controlling gene is present. If a plant inherits the allele for a dominant trait from either parent, that trait will be visible in its appearance. Recessive traits, on the other hand, need both copies of the controlling gene to be the same before they can show themselves. If only one copy is recessive and the other is dominant, the dominant trait will mask the recessive one. In Mendel's garden, this could be seen in the color of the pea plant's flowers. He crossed plants with purple flowers and plants with white flowers. In the first generation of offspring, every plant had purple flowers. The trait for purple flowers was dominant, while the trait for white flowers was recessive. The recessive trait did not disappear, it was simply hidden. When Mendel allowed these purplely flowered plants to self-pollinate, the white flower trait reappeared in the next generation, in a predictable ratio. This was a revelation. It meant that traits could be carried silently through one generation and reappear in the next. A plant or even a person could hold within it the instructions for a trait that was not visible on the surface. These hidden instructions could pass quietly from parent to child until the right combination allowed them to emerge again. The same principle applies to many traits in humans and other animals. For example, in certain cases of eye color, the allele for brown eyes can be dominant over the allele for blue eyes. A person with one brown eye allele and one blue eye allele will typically have brown eyes, though they still carry the blue eye instruction, which could appear in their children if matched with another blue eye allele. It's important to remember that dominance does not mean better or stronger in a practical sense. It is simply a way of describing how traits are expressed. A dominant trait is not necessarily more common in the population, nor is a recessive trait necessarily rare. These terms describe the relationship between alleles which trait is visible when two different versions of a gene are present. Some traits follow these simple patterns closely, but others are more complex. In some cases, neither allele is completely dominant over the other. This can lead to a blending of traits, as in the pink flowers of snapdragons that result from crossing red-flowered and white-flowered plants. In other cases, both alleles can be expressed equally, a situation called codominance, as in certain blood types in humans. Mendel's careful observations of dominant and recessive traits helped scientists understand why certain features seem to skip generations. For example, a recessive trait like attached earlobes in humans might not appear in a child if only one parent carries the allele. But if both parents carry it, even if neither shows the trait themselves, it has a chance of appearing in their offspring. Dominance and recessiveness are not limited to physical traits like color or shape. They can also influence less visible characteristics, such as the way enzymes work in the body or how cells respond to certain chemicals. These hidden traits may not be obvious in everyday life, but they can play a role in health, metabolism, and other internal processes. Mendel's work revealed a beautiful truth. The genetic story is not always visible on the surface. Some traits are bold and present, shaping what we see. Others are quiet and hidden, waiting for the right moment to appear. Together they form a rich tapestry of possibility, ensuring that life remains diverse and full of variation. Even in a quiet garden, the principles of dominance and recessiveness are at work all around us. In the color of petals, the texture of seeds, the length of stems traits are being expressed, hidden, and passed along according to the simple yet profound rules Mendel uncovered. These rules help explain the endless variety we see in nature, even among individuals of the same species. Understanding dominance and recessiveness also helps us appreciate the complexity of heredity. While the basic rules are simple, their combinations and interactions can produce an almost infinite range of outcomes. This is why no two people, plants, or animals are exactly alike, even identical twins, who start with the same genetic code, can develop small differences over time due to environmental influences. In the next part of our journey, we will explore how changes in the genetic code mutations can introduce new variations into this intricate system. These changes, while sometimes subtle, have the power to shape the traits we see and the hidden traits we carry. But for now, we can rest in the understanding that in the quiet conversation between dominance and recessiveness lies much of the visible beauty and hidden potential of life. 12. The role of mutations. If we imagine the genetic code as a book, a vast and ancient manuscript written in the four-letter alphabet of DNA, then a mutation is a change in that text. It might be as small as replacing a single letter with another, or as large as adding or removing entire sections. Some changes are so subtle that they barely alter the meaning of the instructions. Others can shift the story in ways that are profound. Mutations are a natural part of life. They occur whenever DNA is copied, whether during the formation of new cells in a growing body or in the passing of genetic information from one generation to the next. DNA replication is an extraordinarily accurate process, but it is not perfect. From time to time, an incorrect base is inserted, a section is skipped, or a strand breaks and is rejoined differently. These changes can happen for many reasons. Some arise spontaneously, simply as a byproduct of the complex chemistry of life. Others are triggered by external influences, sunlight, cosmic rays, naturally occurring chemicals in food or water, or even the normal byproducts of metabolism inside the cell. The DNA molecule is strong, but it is not immune to these subtle forces. Fortunately, living cells have repair systems, intricate molecular tools that constantly scan the DNA for errors. These repair mechanisms can recognize a mismatched base, snip it out, and replace it with the correct one. They can rejoin broken strands, mend chemical alterations, and restore the original structure. Without these repairs, the accumulation of mutations would quickly overwhelm the delicate balance of life. Still, not every change is corrected. Some mutations remain, quietly becoming part of the genetic record. Many of these have little or no noticeable effect. The change may occur in a part of the DNA that does not code for proteins, or it may alter a protein in a way that does not disrupt its function. These neutral mutations are like small spelling variations in a long text. The meaning remains the same. Some mutations, however, can be harmful. They might alter a protein so that it no longer works properly, or disrupt the regulation of a gene so that it is expressed at the wrong time or in the wrong amount. In certain cases, harmful mutations can lead to genetic disorders or increase the risk of disease. Nature's repair systems and the careful organization of the genome reduce these risks, but they cannot remove them entirely. On the other hand, some mutations can be beneficial. They might change a protein in a way that improves its function, or introduce a new variation that helps an organism survive in its environment. Over generations, beneficial mutations can spread through a population, becoming part of the shared genetic heritage. This is one of the main ways evolution works, through the slow accumulation of small, favorable changes. In nature, beneficial mutations often arise quietly, without fanfare. A change in a flower's pigment might make it more attractive to certain pollinators. A shift in the structure of a protein might allow an enzyme to work more efficiently in a slightly different temperature. A subtle alteration in a bird's wing structure might give it a small advantage in flight. These are the quiet beginnings of adaptation. Mutations are also a source of the variation we explored earlier in our journey. Without them, the genetic code would remain static, unable to adjust to changing circumstances. It is the constant, slow introduction of new variations, most of them small, a few of them meaningful, that allows life to remain resilient over the ages. In humans and other complex organisms, mutations can occur in two main ways. Somatic mutations happen in body cells and affect only the individual in which they occur. They are not passed on to offspring. Germline mutations, on the other hand, occur in the reproductive cells, the sperm or the egg, and can be inherited by the next generation. It is these germline mutations that become part of the continuing story of a species. Sometimes, mutations can be large-scale changes. A section of a chromosome might be duplicated, deleted, inverted, or even exchanged with a section from another chromosome. These structural changes can have significant effects, sometimes disrupting important genes or altering the way they are regulated. At other times, they can create entirely new combinations of genes, introducing possibilities that did not exist before. Mutations remind us that the genetic code is not a frozen document. It is a living text, copied again and again, over countless generations. Each copy is almost perfect, but never entirely the same. Over time, these differences accumulate, shaping the course of life. Without mutation, evolution would stand still. With it, life can explore an infinite number of paths. When we think of mutations, it can be tempting to focus only on the harmful ones, as they can have immediate and visible effects. But in the broader perspective of life, mutation is neither purely good nor purely bad. It is simply change, and change is the raw material from which nature builds diversity. We can think of mutation as the quiet hand that occasionally edits the manuscript of life. Most edits are small, leaving the meaning intact. Some create new possibilities. Some, though rare, may cause problems. But without any edits at all, the story would never grow, never adapt, never branch into the countless forms of life we see today. In the next part of our journey, we will explore how the instructions in DNA, whether old, new, or altered by mutation, are put to use in the body. We will see how genes are read, how their information is turned into proteins, and how those proteins bring the genetic code to life. But for now, we can rest in the awareness that the very changes that make each living thing unique often begin as the smallest of shifts in the most delicate of codes, a single letter changed, a single thread of the spiral altered, quietly adding to the great unfolding story of life. 13 DNA to protein, the work of cells. Every cell in your body contains the complete genetic code, yet not every cell uses the same parts of that code at the same time. The genes inside DNA are like instructions in a vast library, but instructions alone do nothing unless they are read and acted upon. In the quiet, hidden world inside the cell, this reading and acting is the process by which DNA becomes protein, the transformation of written code into the very tools and structures of life. Proteins are the workhorses of biology. They are builders, messengers, protectors, and catalysts. Some give structure to cells, forming scaffolds that hold everything in place. Others act as enzymes, speeding up chemical reactions that would otherwise take far too long to sustain life. Still, others are messengers, carrying signals from one part of the body to another. The diversity of proteins is immense, and each one is shaped by the precise sequence of nucleotides in its corresponding gene. The journey from DNA to protein begins with transcription. This process takes place in the nucleus, the cell's secure archive where DNA is stored. A section of DNA containing a gene unwinds and separates, revealing the sequence of bases on one strand. An enzyme called RNA polymerase moves along this strand, reading each base and building a complementary strand of RNA. This RNA copy is called messenger RNA, or mRNA. Messenger RNA is similar to DNA, but it has a few differences. It uses the base uracil, U, in place of thymine, T, and it is a single strand rather than a double helix. The mRNA carries the genetic information in a form that can leave the nucleus and travel to the protein-making machinery of the cell. In a way, it is like checking a reference book out of a library. The original stays safely inside, while a copy goes out into the world to be used. Once the mRNA is complete, it leaves the nucleus through small openings in the nuclear membrane and enters the cytoplasm, the fluid-filled space of the cell. Here, the mRNA finds the ribosome, a remarkable molecular machine that builds proteins. The ribosome reads the mRNA in sets of 3 bases at a time. Each 3-base set called a codon, corresponds to a specific amino acid, the building blocks of proteins. As the ribosome moves along the mRNA, transfer RNA molecules, tRNA, bring the correct amino acids to match each codon. The ribosome links these amino acids together, one after another, forming a chain. The sequence of amino acids in the chain is determined entirely by the sequence of codons in the mRNA, which in turn comes directly from the DNA. Once the chain is complete, it folds into a specific three-dimensional shape. This folding is essential because the shape of a protein determines how it works. Some proteins fold into long, strong fibers that provide support and structure. Others fold into compact, intricate shapes that fit perfectly with other molecules, allowing them to carry out precise chemical reactions. This entire process, transcription in the nucleus, translation at the ribosome, folding into functional form, is known as gene expression. It is how the silent instructions in DNA become the active components of life. The process is tightly regulated. Not every gene is expressed all the time. Cells turn genes on and off, depending on their needs and their environment. For example, a skin cell will express genes that make structural proteins like keratin, which gives skin its strength, but it will not express the genes needed for producing the digestive enzymes found in the stomach. This selective reading of the genetic library allows each cell type to specialize while still carrying the same complete set of instructions. Gene expression can also respond to changes in the environment. For example, certain bacteria will switch on genes for digesting a new food source when it becomes available. Plants can adjust the expression of genes related to photosynthesis depending on the amount of sunlight they receive. Even in humans, the expression of certain genes can be influenced by diet, stress, temperature, and countless other factors. The elegance of this system is that the DNA itself rarely leaves the safety of the nucleus. Instead, it sends out copies mRNA molecules that can be read, used, and then broken down when they are no longer needed. This protects the original code from damage, while still allowing the cell to use the information whenever required. Proteins themselves are not permanent. They are constantly being built, used, and replaced. Some proteins last for only a few minutes before being recycled, while others may endure for weeks or months. The constant renewal ensures that the cell's machinery stays fresh and responsive. The relationship between DNA and protein is the central thread of biology. It is the bridge between the genetic instructions we inherit and the living, functioning organism we become. Without this process, the genetic code would remain silent, a library of unopened books. With it, the code becomes alive, building the countless structures and systems that make life possible. We can imagine this process as an endless, quiet conversation between the nucleus and the ribosomes. The nucleus says, "Here is the plan," and the ribosomes respond, "We will build it." The result is the constant renewal of life, carried out silently in every cell, every second of every day. In the next part of our journey, we will explore how genes and the environment interact, how our inherited code works in partnership with the world around us to shape who we are. But for now, we can rest in the knowledge that the genetic code is not only written and stored, it is read, interpreted, and put into action, creating the very fabric of life from the smallest molecular details to the grandest forms in nature. 14. The dance of nature and nurture. Every living being carries within it a unique genetic code, a quiet script written long before birth. This code shapes much of who we are, the architecture of our bodies, the way our cells work, even some of our tendencies and abilities. But our genes are not the only influence on the unfolding story of our lives. Around and alongside them is the environment, the world we live in, the conditions we experience, the choices we make, and the subtle influences we may never fully see. This meeting point between our inherited code and our lived experience is often called the interplay of nature and nurture. Nature refers to the traits and tendencies that come from our genes, while nurture describes the influences of our environment. Together, they weave the tapestry of life, each thread strengthening, softening, or shaping the others. Some traits are strongly influenced by genetics. The color of a flower's petals, the pattern of a tiger's stripes, or the basic structure of a bird's wing are all written into the genetic code. These traits will appear much the same regardless of the environment, because the instructions for them are stable and precise. But many other traits exist in a more delicate balance. They emerge from the ongoing conversation between the code within and the world without. Take human height as an example. Genes play a significant role, providing the blueprint for how tall a person can grow. But nutrition, health during childhood, and other environmental factors can influence whether that potential is fully reached. A person whose genes allow for tall stature may grow shorter if they lack proper nourishment, while someone with a naturally smaller genetic frame may grow taller if their environment strongly supports healthy growth. The environment's influence can be subtle and long-lasting. Temperature, light, and available food can change how genes are expressed not by altering the DNA sequence itself, but by influencing which genes are turned on or off. In plants, for instance, sunlight can affect the production of pigments in leaves and flowers, changing their color intensity. In animals, changes in diet can influence metabolism and body composition, even when the genetic code remains the same. Scientists now understand that this influence works in part through a process called epigenetics. Epigenetic changes are like bookmarks placed in the genetic text, marking which sections should be read more often and which should be read less. These marks can be added or removed based on life experiences, and in some cases, they can even be passed to future generations. This means that nurture, the influence of the environment, can leave its own quiet traces in the story of life, shaping not only an individual, but also the generations that follow. The dance between nature and nurture can also be seen in behavior and learning. Songbirds, for example, are born with the instinct to sing, but the exact song they sing is learned from hearing adults. Their genes give them the ability to sing. Their environment teaches them the melody. Humans, too, have many abilities shaped in this way: language, movement, creativity. Our genetic potential sets the stage, and our experiences fill in the details. Sometimes, the environment can reveal hidden traits. A plant might carry the genetic ability to grow deep roots, but it will only do so if the soil becomes dry and water is scarce. A person might have a genetic tendency for endurance, but that trait will only flourish if they live in a way that develops their stamina. In these cases, the environment acts as a quiet key, unlocking certain possibilities within the genetic code. The reverse can also happen. The environment can soften or suppress traits that might otherwise be strong. Stress, poor health, or lack of opportunity can limit the expression of certain abilities, even when the genetic potential is present. This is part of why the same genes can lead to different outcomes in different people, depending on the conditions of their lives. Understanding the balance between nature and nurture is not about deciding which is more important. It is about seeing that they are inseparable partners. Genes provide the foundation, but the environment shapes the structure built upon it. Without the blueprint, the structure could not exist. Without the shaping, the blueprint would never become reality. In humans and other animals, this interplay is constant and lifelong. Our genes influence how we respond to our environment, and our environment influences how our genes are expressed. It is a feedback loop, a gentle back and forth, that continues from the earliest stages of development until the end of life. This perspective reminds us that our story is both inherited and created. We carry the echoes of those who came before us in our DNA, but we also write new chapters through the lives we lead and the choices we make. The world we live in, the people we meet, the experiences we gather, all leave their marks alongside the marks of our genes. Nature and nurture do not compete. They move together, each adding to the other. Like two dancers, they step in rhythm, sometimes one leading, sometimes the other, but always moving as a pair. And it is from their dance that the full expression of life emerges, not fixed, but ever changing, shaped by both the quiet code within and the living world around it. In the next part of our journey, we will turn our attention to how modern science works with this delicate balance. We will see how genetics is used today in medicine, in agriculture, in conservation, and how these uses reflect our growing understanding of the partnership between inherited potential and the influence of the environment. But for now, we can rest in the awareness that we are not shaped by one or the other alone. We are the product of both of nature's quiet blueprint and nurture's guiding hand, working together in the endless dance of life. 15. Modern genetics and daily life. In the quiet of our everyday lives, it is easy to forget that genetics is not just a distant science studied in laboratories or written about in textbooks. It is woven into the fabric of our daily experience, quietly shaping the world around us and the choices we make. Modern genetics is not a separate realm of knowledge. It is part of the food we eat, the medicine we take, the conservation of the natural world, and even the understanding of our own personal stories. In medicine, genetics plays a central role. Doctors now use genetic information to better understand certain illnesses, to predict risks, and to choose treatments tailored to the individual. This approach, often called personalized medicine, recognizes that the same illness can behave differently in different people because of variations in their genetic code. By studying these differences, doctors can choose treatments that are more likely to work for a specific patient and less likely to cause side effects. For some conditions, genetic testing can reveal whether a person carries a certain variant that increases their risk. This knowledge can guide preventive measures, encouraging lifestyle choices or monitoring that might reduce the likelihood of illness. In other cases, genetic testing can help confirm a diagnosis, especially for rare conditions that might otherwise be difficult to identify. Genetics has also transformed our understanding of infectious diseases. By studying the genetic code of viruses and bacteria, scientists can track how they spread and change over time. This information helps in designing vaccines, developing new treatments, and understanding why some people are more resistant to certain infections than others. In agriculture, genetics quietly shapes the food we grow and eat. Farmers have long selected plants and animals for desirable traits: sweeter fruit, higher yields, better resistance to pests. Today, genetic science allows us to understand these traits more precisely. By studying the DNA of crops and livestock, scientists can help breeders develop varieties that are more nutritious, more resistant to disease, and better able to grow in changing climates. Genetics also plays a role in preserving biodiversity. Conservation biologists use genetic information to understand the health of wild populations. By studying the genetic diversity within a species, they can identify whether a population is strong enough to survive changes in its environment, or whether it needs help to recover. This genetic knowledge guides breeding programs for endangered species, helping ensure that future generations are strong and adaptable. Even in our personal lives, genetics has found a place. Many people choose to explore their ancestry through direct-to-consumer DNA testing. These tests can reveal where a person's ancestors came from, how populations moved across the world, and sometimes even connect distant relatives. While such tests cannot tell the whole story of a person's heritage, they can open windows into the past, linking individuals to the larger human family. Genetics also influences the way we think about food, exercise, and lifestyle. Some people have genetic variations that affect how they metabolize certain nutrients or how their bodies respond to physical activity. While the science of applying this information to daily life is still developing, it is part of a growing understanding that our needs are not one exa, fiasa. Modern genetics also touches on questions of ethics and responsibility. As our ability to read and even edit the genetic code grows, we must consider how we use that power. Techniques like CRISPR allow scientists to make precise changes to DNA, offering possibilities for curing genetic diseases or improving crops. But they also raise questions about where to draw the line about what changes are wise, what changes are safe, and what changes we should avoid. In this way, modern genetics is both a tool and a mirror. It allows us to change the world around us in ways that were once unimaginable, and it reflects back our values and priorities as a society. The choices we make about how to use genetic knowledge will shape not only the future of science, but the future of life itself. It is also important to remember that while genetics can offer powerful insights, it is not the sole answer to every question. Genes are only part of the story. Environment, experience, and chance all play their parts. A genetic predisposition is not a destiny, it is one factor among many, and understanding it can help guide, but not dictate, our lives. For most of us, the influence of modern genetics is quiet and indirect. We may not think about it when we take a medicine, choose a fruit at the market, or read about conservation efforts for an endangered species. Yet it is there, shaping the decisions behind the scenes, helping create a world that is more responsive to the needs of individuals, more protective of biodiversity, and more capable of meeting the challenges of the future. Modern genetics is, in many ways, an extension of the same principles that Gregor Mendel discovered in his pea garden. It is still about understanding how traits are passed on, how variation arises, and how life adapts. But today, it is also about applying that understanding in countless practical ways in clinics, on farms, in conservation programs, and even in our homes. In the next part of our journey, we will reflect on how this science connects us not just to our own lives, but to the larger story of all living things. We will explore how understanding genetics can deepen our sense of belonging to our families, our species, and the vast web of life that covers the earth. But for now, we can rest in the awareness that modern genetics is not a distant or abstract idea. It is here, all around us, quietly shaping the world we live in every day. 16. Understanding ourselves through genetics. To understand genetics is to understand that each of us carries a story far older than we are. It is a story written not in ink, but in the spiraling language of DNA, passed quietly from generation to generation. Every cell in our body holds a complete copy of this story, a record of our biological heritage that stretches back through countless ancestors, connecting us to people and places we may never know by name. Our genes are not simply instructions for building a body, they are a bridge between past and present, linking us to the people who came before us and to the shared history of humanity. They carry the echoes of survival, of adaptation, of choices made not by us, but by those whose lives shaped the path that led to ours. When we look closely at our genetic code, we see not just ourselves, but the long chain of life we belong to. Modern science allows us to read parts of this code in ways that our ancestors could never have imagined. Genetic studies can reveal where our family lines once traveled, which regions of the world our ancestors called home, and even which ancient populations contributed to our genetic makeup. This knowledge does not replace the lived traditions and cultural histories that form our identities, but it can enrich them, adding another layer to the tapestry of who we are. Beyond ancestry, our genetics can also tell us something about how our bodies work. Certain genes influence how we process nutrients, how we respond to exercise, or how susceptible we may be to specific illnesses. Understanding these genetic influences can help us make informed choices about our health, while also reminding us that we are not bound entirely by our DNA. The environment we live in, the food we eat, the activities we choose, and the care we give our bodies can all influence how these genetic tendencies are expressed. One of the most profound insights genetics offers is the realization of how much we share with one another. While the small differences in our DNA help explain the variety we see in human beings, the similarities are far greater. In fact, all humans are more than 99% genetically identical. That remaining fraction of difference accounts for everything from hair texture to eye color to subtle variations in metabolism. This shared genetic foundation is a reminder that, beneath the surface, we are all part of the same extended family. Genetics also reveals our connection to the rest of life on Earth. Many of our genes are similar, in some cases nearly identical to those found in other animals, from primates to fish to even the simplest of single-celled organisms. These shared genes are evidence of a common origin, a sign that all living things are branches of the same ancient tree of life. To understand this is to see ourselves as part of nature, not separate from it. Learning about our own genetics can be a deeply personal experience. It can explain traits we've noticed in ourselves or in our families. It can reveal hidden connections to people and places we've never met. And sometimes, it can bring unexpected discoveries, a trait we carry that we didn't know about, a link to an ancestor we had never heard of, or a shared sequence of DNA with someone far away. But understanding ourselves through genetics is not only about looking inward, it is also about recognizing the bonds we share with others. Every gene we carry has a history, shaped by migrations, by adaptation to climates, by survival, through challenges that stretch far beyond our own lifetimes. Our genetic code is a record of humanity's collective journey, and every person alive today carries a part of that shared record. This knowledge can also deepen our sense of empathy. To see how closely related we are, genetically and biologically, is to see that the divisions we draw between ourselves are small compared to what unites us. The differences that seem large on the surface are just variations within a single interconnected species. In this way, genetics can remind us of our common ground and perhaps inspire us to treat one another with greater kindness. Understanding ourselves through genetics is also about appreciating the balance between what we inherit and what we create. Our DNA provides the starting point, the foundation on which our lives are built. But how we live, the choices we make, and the environments we inhabit add to and shape that foundation. We are not simply the sum of our genes. We are the living result of the interplay between our genetic heritage and our lived experience. In the stillness of reflection, we might imagine our genetic code as a quiet river flowing from the past into the present. The river carries the waters of many streams, parents, grandparents, ancestors, long forgotten, and it continues on into the future, carrying part of us forward into generations yet to come. We are both a point along its course and a part of its ongoing journey. In the next part of our journey, we will look outward from the self and consider the larger picture, the shared thread of genetics that connects all living things. We will see how this common heritage weaves together the tapestry of life on Earth, reminding us that we are part of something far larger than ourselves. But for now, we can rest in the awareness that in every cell of our being lies a story that began long before we were born, a story that is still unfolding, and one that will continue long after we are gone. 17. The common thread of life. If we look closely at the world around us, at the forests, the oceans, the open skies, we see life in a breathtaking variety of forms. Towering redwoods, delicate orchids, soaring eagles, swift cheetahs, tiny plankton drifting in the sea. At first glance, these forms seem completely different from one another, each shaped by its own unique path through time. But beneath their outward differences lies something remarkable, a shared foundation, a single thread that ties all living things together. That thread is the genetic code. Every living organism on Earth stores its instructions in the same molecular language, DNA. From the smallest bacterium to the largest whale, the genetic code uses the same 4 letters A, T, C, and G to spell out the recipes for life. The sequence of these letters may vary from species to species and from individual to individual, but the language itself is universal. It is as though all of life is written in a single alphabet, rearranged into countless stories. This shared code is evidence of a common origin. Billions of years ago, the first simple life forms emerged on Earth, carrying with them a primitive version of the DNA-based system we know today. Over vast stretches of time, these early organisms diversified, adapting to new environments, evolving into new shapes and forms. Yet through all these changes, the core language of DNA remained the same. It is the oldest unbroken tradition in the history of life. The similarities in our genetic code reveal connections we might never expect. Humans share around 99% of our DNA with chimpanzees, our closest living relatives. We share about 85% with mice, 60% with fruit flies, and even a surprising 40–50% with certain plants. These shared sequences are not random. They are the inherited remnants of ancient ancestors, preserved because they work so well in building and sustaining life. In a sense, we are all variations on a theme. The differences between species come from how the genetic instructions are arranged and used, not from an entirely separate blueprint. The same basic genetic tools are reshaped again and again, giving rise to the endless variety of life we see today. This common thread connects us not only to animals and plants, but also to the smallest forms of life. Microorganisms, bacteria, archaea, and microscopic algae carry DNA that is structured in much the same way as ours. In fact, many of the essential processes that keep our cells alive copying DNA, building proteins, generating energy, are shared across all domains of life. These processes are so effective that they have remained largely unchanged for billions of years. Understanding this shared genetic heritage changes the way we see the natural world. It reminds us that the boundaries we draw between species are, in some ways, human-made distinctions layered over a much deeper reality. Life is not a collection of isolated forms. It is a single, vast family, with each branch connected to the others by the thread of DNA. This perspective can also deepen our sense of responsibility toward other living beings. When we recognize that we share so much of our genetic foundation with the creatures around us, it becomes harder to see them as wholly separate from ourselves. The genetic kinship we have with a bird in the sky, a tree in a forest, or a fish in the sea is a reminder that the fate of each species is intertwined with the fate of the whole. In conservation biology, this understanding is more than philosophical. It is practical. Protecting biodiversity means preserving the genetic diversity within and between species. Every species carries unique genetic variations, shaped by millions of years of adaptation. When a species is lost, an entire branch of the genetic tree disappears with it, and the thread of life becomes a little thinner. Even in our daily lives, the common thread of DNA has quiet effects. The foods we eat, the medicines we take, and even the air we breathe are the result of interactions between countless species, all bound together by the same underlying genetic machinery. The oxygen in the air, for example, is produced by plants and photosynthetic microorganisms distant relatives of ours, whose genetic heritage makes them capable of turning sunlight into the energy that sustains the planet. It is humbling to realize that every living thing you see, every leaf, every bird, every person, is carrying forward the same ancient tradition. Each organism is a new chapter in the ongoing story of life, written in the same universal code, with variations shaped by countless generations before. In moments of reflection, we might imagine life as a great woven fabric, stretching across time and space. The threads are many, but they are spun from the same fiber. The patterns shift, grow, and change, but the underlying material is constant. Each organism is a knot in that fabric, connected to others by the same enduring thread. In the next part of our journey, we will turn from this shared foundation to consider how genetics not only shapes life as it exists now, but also guides its unfolding into the future. We will explore how the ongoing changes in DNA through mutation, selection, and adaptation keep the thread of life moving forward, weaving new patterns for the generations to come. But for now, we can rest in the awareness that no matter how different we may seem, all living things are bound together by the same quiet, ancient code, the common thread of life. 18. The future written in our genes. The genetic code is often thought of as something ancient, a record of the past, a link to the lives of those who came before. And it is. Every twist of DNA carries echoes of survival, of adaptation, of challenges met by generations stretching back into deep time. But DNA is not only a reflection of the past, it is also a blueprint for the future, a set of instructions that will be passed on, carried forward, and reshaped by the lives yet to come. When we speak of the future written in our genes, we are speaking of potential. Our DNA holds possibilities, some visible, some hidden, that may be expressed in new and unexpected ways as life continues. It is a quiet storehouse of traits, variations, and abilities that have been shaped by countless generations and will continue to evolve. This evolution happens through the same gentle processes we have explored: mutation, selection, and adaptation. Mutations introduce new variations into the genetic code. Most of these changes are small and subtle. Some will be neutral, a few may be harmful, and a rare few will be beneficial. Natural selection acts on these variations, favoring those that help an organism survive and reproduce in its environment. Over time, these favored traits become more common, shaping the direction of a species' development. In this way, the future of life is written in an ever-changing script. Even as the genetic code remains deeply familiar, the same 4 letters, the same basic structure, the order of those letters shifts slightly over generations, creating new patterns. The differences may be so small that they are invisible in a single lifetime, yet over centuries and millennia, they can transform species. Humans are no exception. Our own species continues to evolve, even if the changes are less obvious than in the wild animals and plants around us. Some adaptations are responses to our physical environment, such as variations in skin pigmentation, shaped by exposure to sunlight in different parts of the world. Others are responses to cultural and dietary changes, such as the ability to digest milk into adulthood in populations that domesticated dairy animals. The future of our species will also be influenced by the choices we make today. Advances in medicine, technology, and agriculture may alter the kinds of traits that are favored. Our ability to travel, to share information instantly, and to live in environments vastly different from those of our ancestors changes the pressures that once shaped human evolution. Climate change, shifting ecosystems, and the movement of people and cultures will all influence which genetic variations are passed on. In the broader living world, the future written in genes will be shaped by both natural and human-driven changes. As habitats shift, species will adapt or attempt to adapt to new conditions. Some will succeed, carrying forward variations that help them thrive in the altered world. Others may struggle, their genetic diversity too narrow to keep pace with change. Conservation efforts can help by protecting habitats, supporting genetic diversity, and giving species the chance to adapt. There is also another layer to the future of genetics, one that is new in the history of life. For the first time, humans have the ability to read, copy, and even alter the genetic code with precision. Technologies such as CRISPR allow scientists to make specific changes to DNA, correcting harmful mutations or introducing traits that might never have arisen naturally. This ability holds great promise: the potential to cure genetic diseases, to protect crops from pests without chemicals, to help species adapt to environmental changes. But with this power comes responsibility. Altering the genetic code is not the same as reading it. The changes we make today could echo far into the future, carried forward in the same way that natural variations are passed from generation to generation. The challenge is to use this knowledge wisely, to weigh the benefits against the risks, and to remember that the genetic story is part of a much larger web of life. The future written in our genes will also be shaped by the balance between stability and change. Too much change, too quickly, can be disruptive. Too little change can leave a species unable to adapt. Life has always moved between these two forces, adjusting its pace according to the challenges it faces. This is the rhythm that has kept life thriving for billions of years. When we think about our own place in this future, it can be humbling to realize that each of us is both a product of the genetic past and a contributor to the genetic future. The traits we carry, the choices we make, the environments we create, all will have ripples that extend far beyond our own lifetimes. The genes we pass on, whether to children, to other living things we influence, or simply to the shared pool of life on Earth, will help shape what comes next. We may not be able to predict the exact form the future will take, but we can know this: the thread of life will continue, woven from the same ancient fibers, carrying forward both the old patterns and new ones yet to be imagined. The story will be told in the same language, but with chapters still unwritten. In the next and final part of our journey, we will step back and reflect on what we have learned not only about the science of genetics, but about what it tells us of our place in the world, our connection to one another, and the quiet, enduring story of life that we are all a part of. But for now, we can rest in the knowledge that our genes are not just relics of the past. They are seeds of the future, carried within us, ready to take root in generations yet to come. 19 closing reflections. The endless story of life. We have traveled together through the winding pathways of genetics, from the smallest building blocks of DNA to the vast web of life that binds all living things. Along the way, we have seen how the instructions for life are written, how they are passed down, and how they change, adapt, and grow through time. Now, at the close of our journey, we can pause to reflect on what this story tells us about ourselves, our place in the world, and the continuity of life itself. Genetics begins with something so small it is invisible to the eye, a code made of just 4 chemical letters, arranged in countless patterns. Yet from this simple alphabet, life writes endless variations. The same code builds the shell of a snail, and the trunk of an oak tree, the wing of an eagle, and the human hand. It is a shared language, spoken fluently by every living thing, passed unchanged in its essence from the dawn of life to this very moment. Within that code lies both memory and potential. Memory, because each sequence of DNA carries the history of survival, the adaptations that worked, the solutions that allowed life to endure. Potential, because the code is not fixed in stone. It can change, gently and gradually, allowing life to meet new challenges and to flourish in new forms. The story of genetics is also a story of connection. Every human being carries a unique version of the genetic script, but that uniqueness is written on a foundation we all share. We are more alike than different, more connected than separate. The variations that make each of us an individual exist within the larger unity of the human family, and within the still greater unity of all living things. It is humbling to realize that your DNA is not entirely your own. It is an inheritance, carried to you by parents, grandparents, and countless ancestors whose names you will never know. It is a gift shaped by their lives, their environments, their struggles and triumphs. You are, in a very real sense, the latest chapter in a book that has been writing itself for billions of years. This perspective can change how we see the world. When we look at a tree, we can know that somewhere deep in its cells lies the same universal code that is in ours. When we hear the song of a bird, we can know that the instructions for its voice are written in the same alphabet as the instructions for our own speech. This is the common thread the quiet truth that all life is related. The science of genetics gives us tools to understand this truth more clearly. It shows us how traits are inherited, how variations arise, how species adapt to their surroundings. It explains the mechanisms behind the beauty of a flower, the resilience of a migrating bird, the endless diversity of life on Earth. But beyond the science, genetics offers a deeper reminder that life is one long, unbroken story. Our part in this story is both temporary and eternal. Temporary, because each individual life is brief, a single moment in the long river of time. Eternal, because the genetic thread we carry does not end with us. It flows onward, woven into the lives that follow, just as it was woven into ours from the lives that came before. In this sense, genetics is not only about understanding life, it is about belonging to it. It is about recognizing that we are part of something vast, intricate, and enduring. Every breath we take, every cell in our body, every heartbeat is part of a chain of life, stretching back to the very beginning and forward into the unknown future. The knowledge of this connection can bring a kind of peace. It reminds us that even when our own chapter ends, the story will continue written in the same alphabet, shaped by the same forces, carried by the same thread of DNA. Life will keep telling its tale, just as it always has, with new variations, new adaptations, new expressions of the same ancient code. And so, as we reach the end of our exploration, we find ourselves not at a conclusion, but at a continuation. The story of genetics has no final chapter. It is an open book, still being written in every seed that sprouts, every egg that hatches, every child that is born. We are not only readers of this story, we are characters in it, and in small ways, we are also its authors. In the quiet of this moment, we can imagine that story stretching out before us, a tapestry woven from countless threads, shimmering with the light of all the lives that have ever been, and all those yet to come. Each thread is unique, yet all are part of the same fabric. And if we listen closely, perhaps we can hear the soft whisper of life itself, speaking in the language of DNA. I was here before you, I am here with you now, and I will be here long after you are gone. The endless story of life is one we all share. It is written in our cells, carried in our blood, whispered in our breath. And though the details change, the theme remains the same: survival, adaptation, connection, and the ceaseless unfolding of possibility. This is the gift of genetics, and it is the gift of life itself.